Yeah, I'm going to share with you guys a little bit of the work that we've been doing in Cortical Labs. Cortical Labs is a Melbourne-based startup that is looking at how can we culture intelligence? In other words, how can we use brain cells in a dish as a type of information processing device? And so, yeah, I'm Brad. I've got a background in neuroscience and stem cell therapy, a bit of regenerative medicine, bioinformatics, quite a broad generalist, I would say. But then we also have an amazing team that we work together to get this work done. And so what exactly is it that we're looking at? We can look at the brain, and I think it's not too big a claim to say that the brain is really, at the end of the day, the most advanced learning system possible. As amazing as something like ChatGPT or Stable Diffusion is at doing what it does, they're not generalized intelligence. They can operate in a very narrow parameters of task. And of course, at the end of the day, a lot of what we see in, say, machine learning, in any case, has been inspired by the brain and what people are trying to model how the brain works. We sort of raised the question, why would you model what you can harness? We know that these biological neurons display an innate learning ability, and you see that across all scales, from flies to dogs to humans. They all use biological neurons as this basis. And when they form parts of the brains, they can do a number of different tasks, and they do so with incredibly low power consumption and can maintain that function even when they suffer sometimes catastrophic damage, which is very exciting to see as a viable system we could harness. And so how would we get there, though? Obviously, this is not exactly a trivial process, and it requires you to bring together a lot of different expertise and fields if you really want to get out there and harness brain cells at their sort of substrate fundamentals. And so what we aimed to do was combine advances in hardware, software, neurocomputational understandings, and synthetic biology, and try to bring all of those together. So what we can do is we can have improved hardware and software to allow for a real-time closed-loop implementation. And this is something we call an embodied network. We can have more detailed theories of how neural systems actually optimize their behavior for intelligence. And one of the theories we tested was actually called the free energy principle. And then synthetic biology techniques can also be used to actually generate the cells in a scalable and ethical way to develop, let's say, specific cell types, such as cortical neural networks. So I'll just touch on some of those terms incredibly quickly, especially with the time. So an embodied neural network is essentially something that we have in a closed loop feedback. What closed loop means is that you have to stimulate a sensation into, let's say, this network of biological neurons in a dish. You have to measure the response, and then you have to provide future sensory feedback on how that response is impacted in the world by altering the future sensation. Now, this should ideally be high resolution, real time, and physiologically relevant. And then by doing so, essentially what you create is this barrier between the internal and the external system, or what we would often call a body, in terms that there is a divide between the inside and outside. So it's not some sort of sense that there's some sort of consciousness or anything else like that going on here. It's simply a way to statistically say that there is a barrier between the internal and external world in terms of the information flow. That's what we call embodiment. And we know that this is incredibly important. Studies looking at it where they disrupt the closed loop system, let's say in animals, by putting a VR system on them like Attinger did in 2017, and the disconnecting the movement from the feedback shows really stark disruptions to the animals. And we can imagine also for ourselves, we didn't receive feedback about how our actions changed our place in the world and the world around it. It would be very hard for us to exist in that way. However, even if we can interact with a group of neurons in a dish, how do we actually convince them to do anything useful? There is no predisposition for us. We're interested in the question of, can we get them to play a simple game of pong? Spoiler alert, these are actual neurons moving that paddle on the right side. But how and why would they do that? We needed a theory to actually drive that learning. And so we proposed this using this principle. We didn't propose the principle of the free energy principle. Far more clever people, led by Carl Friston over in UCL, as well as many collaborators around the world, have developed this theory, we'd called the free energy principle. And essentially what it proposed was that a neural system would try to predict its environment and align a perception with an observed outcome. Now, if this was true, essentially, And what the system was trying to do is measure a difference between an internal and an external world to minimize what's called the free energy or the information entropy, also referred to as surprisal. If we gave the neural system a non-predictable random stimulation as an outcome of it missing the ball, something we didn't want, but is otherwise perhaps arbitrary to the neural system, they might find that aversive and avoid doing things that led to that. And conversely, predictable stimulation might be considered reinforcing because it's something that's easier to align into the internal models. And so we thought if we did this, perhaps the neurons would change their activity and actually get better at playing the game of Pong as an outcome of them trying to minimize their free energy. So we then also finally cultured cells. I'm not going to go into this in a huge amount of detail, but in short, we took human iPSC cells and we could turn them into cortical neurons using different methods. The initial work I'll show you uses Gilsmeds, and later work uses a lentivirus-based NGN2 work. Or we could also grow them from mice as a comparison. And we did that because we needed a ground truth of these are 100% biological cortical neurons that no one can dispute, and also just to show that it was a generalizable effect across multiple cell types. And then we plated them onto something called a multi-electrode array. I'll show you what that is in a second. And then you can see here, for those of you familiar with immunocytochemistry, I should say, we were able to stain them for markers of neurons, such as neuron. This green here shows cells that are neurons. The red shows the axons. The purple or pink, depending on screen contrast, shows the dendrites. And then over here, we can look at different markers to see how many of those neurons are actually cortical, and a large proportion of them are cortical or supporting. So greens here showing cortical cells, red is showing the supporting glia, and then the pink, of which there is none, would have shown us the binding cells, but we didn't have the binding cells. And that's what we wanted, because we want a nice stable culture that we can culture long-term on the chip, such as what you've got here. So these are cultures that have survived for more than 6 months. They form these dense interconnected networks, and below it is the multi-electrode array I was telling you about. This is essentially a type of chip that is able to record electrical information from the cells, as well as stimulate in a few points electrical information back to the cells. And so this electrical information in and out forms a bridge, a shared language between the silicon and the biology through which we communicate. And what's really interesting about biological neurons is that they actually will go on to develop their own spontaneous activity, even if you just leave them alone. They just will fire off little pulses as they communicate amongst themselves, doing whatever neurons do. And you can see here, the yellow basically shows the frequency of those stimulations over time for different cell types. And although different cell types all show different time courses, you can see here, this is day 18 before we get nice activity for mouse cells. It's day 88 for human cells with this method, and day 34 for this method. They all do develop quite nicely active networks. And so that means really what we've got here with this approach is that we can develop neural cells in multiple ways to have quite an ethic, and especially with the human cell source method, we can do it in an ethical and sustainable way. And I'll touch on that a bit in the end about why that is. And so we can bring all of these different features together into an integrated setup. And so what does that look like? Essentially, we can grow neurons on top of this chip, and then we can map out an otherwise arbitrary manner relative to the structure of the neurons, a sensory region and motor regions, which down here you can see with counterbalance to make sure that they're not too affected by bias. They stimulate on one side and it fires on one side. It's not affected by that. And of course, we tested and validated that. And then what we can do is when we stimulate it, record the information, change the position of the paddle and how the ball is moving, if it's hit, if it's missed, and its feedback. And we do that in real time to create this embodiment in this simulated environment for these little dish of about 800,000 neurons. These are the 8 electrical stimulation points. And what we do essentially, much like the way our eyes work, we put in stimulation in a way that it represents the position of the ball relative to the paddle in something called a topographic stimulation. And then we can also do a rate coding element to this as well, so that the closer it gets, the faster the stimulation occurs, which is something you can see over here. If you're very familiar looking at these things called raster plots, if not, don't worry about it. Perhaps a bit more of a friendly descriptor of how this works is this visualizer, where it'll flip around in a second, but each little tile here is an electrode routed, and this is the sensory region that I was telling you about. And you can see here, these are all neurons firing. It is at 2 times speed, but down here then are the motor regions. And we're simply recording and reading out what's going on to let it move the paddle. There's no complicated machine learning in this version. It's merely, will the neurons change their activity in response to their stimulation and feedback, and will they learn? And so just because of the sake of time, and I know I was a few minutes late, I'm going to go a little quickly over some of these results and just really focus on the key thing. And the key thing is we ran a bunch of different control methods. Control was testing whether our system alone would lead to learning just in media. In silico would a computational model of this that just drove the paddle with random noise learn. Rest was actual biological neurons, but receiving no information. And as the name suggests, it started out literally as a time when the neurons could rest and cool down because the system did generate heat. And then we had our mouse cortical cells and our human cortical cells. And if, unlike me, you don't glare at heat maps for a significant amount of time, they can be hard to read. But if you just compare the sort of top right hand, which is later minutes and more hits for, say, the control versus the mouse and the human cells, you can see that there's a far lighter colors in the top right side, which corresponds to higher chance of more hits happening later. So you can even just see from here, like quite a nice trend sort of mirroring learning occurring in now cells that are actually playing the game versus cells that are resting and versus other control types. If you prefer box plots, you can see a box plot here showing essentially the same data, where essentially there is no change over time in the first 5 minutes to the last 15 minutes of any of the control groups, and significant changes in both the mouse and human cells. So especially the human cells showed the greatest improvement to where they were hitting the ball about a bit more than they were missing it. Now, if that doesn't sound incredibly impressive and you think you can beat it, I would certainly hope you could, because we're talking about here, as I mentioned, 800,000 cells in a dish receiving incredibly coarse frequency. But if you put that in context, 800,000 cells is about a bumblebee, even though this is a simpler system because they're only flat. It's a monolayer, so it's a flat bumblebee. And if you saw a bumblebee hitting a ball slightly more often than it missed, I think you'd be fairly impressed with the bumblebee. Maybe your bar is to be impressed is higher, but it's still early work, so we'll continue to improve. But what's really nice as well is that this result was replicated over a number of different metrics. So previously, I was just showing you the hit to miss ratio. This here is looking at the number of long rallies greater than 3 in any given time period. And that shows pretty much the same result, same trend. Or you can look at the number of times it initially misses the ball. So an ace, taking the term from tennis here. So it doesn't even hit the ball once. That decreases in the exact same trend as what we observed. And so it's fairly, I think, supportive and robust. And we've cut this analysis in many different ways, and the results come out the same, with some nuances, of course, as you investigated, but it's not like there's only one way to cut this data either. And on top of that, what gives me personally a lot of confidence is that when we look at the functional connectivity, this is an example of just one culture, you can actually see there's like very stark differences in how the cultures and the activity is being organized during gameplay versus rest. And again, in so many different ways and levels. And that gives me a lot of confidence, because if they weren't doing some sort of learning, why you would see this sort of reorganization is hard to explain. So you can see here, for example, when they're playing the game, this shows the functional connectivity. And you can see that it's reorganized very distinctly sort of across these two regions, left and right regions. Perhaps there were more cells on this side than the other side, but nonetheless, this is how they're organized. And the same culture, when you don't stimulate it and you leave it at rest, reorganizes itself like this, much less synchrony between them and connectivity between them. And this is just one quick visual example, I think, too, that we could show you for that, but there are so many more.